Okay, and our next speaker is Moises Exposito Alonso from the Department of Plant Biology at the Carnegie Institute of Science. The title of his talk is The Causes and Consequences of Natural Selection in the Arabidopsis Genome Across Climates. Are you ready? Okay, unmute. Okay.
All right. Do you hear me well and see the slide well? Awesome. Great. So hi, everybody. First, I am humbled to be part of this group of awardees in the honor of Jasper Loftus Hills. So thanks so much for this. And also thanks the organizers for all the work keeping American naturalists connected during these times. So today I'm going to talk, as Susan just said, about how we can use genomics in combination of ecological and evolutionary concepts to understand and predict the evolutionary pressures climate change imposes on a species. So climate change, as you know, is inducing mortalities in many ecosystems, like the massive California fires that we saw last year, longer and extremer droughts threatening plant population persistence. These might be extreme examples that catch our attention, but are the tip of the iceberg of what there are probably many hidden stressors already noticed by species. So when we're trying to conceptualize how a species might be responding to climate change, we typically resource or think of its distribution. So here are cartoons of an idealized distribution of a species in the present with latitude and longitude and a gradient of an environment that might be temperature. And we might think that there are different options for it to respond to a shift in this environmental gradient. One very intuitive is that they will migrate polewards or higher in altitude, tracking their climate niche. And there's plenty of evidence for that. And people have quantified how many kilometers they're moving per decade. And another option would be if they're not able to tolerate this change in climate, there might be some local extinctions and mortalities. And we can see this as a shrinkage of the species distribution. And there's also plenty of evidence for this. What is more difficult to study and see at the species distribution level is if adaptation has happened, as we wouldn't see any change in the shape of the distribution, but arguably this would be optimal because there wouldn't be any loss of biodiversity. There wouldn't be any loss of these populations. The good news, even though it is more difficult to quantify, is that there are many species that are locally adapted to their own, or each population within the species are locally adapted to their own very own local environment. And this might mean that there is genetic standing variation that could be used to catalyze an evolutionary response and adapt to the new climates. So this is the center of my work. How can we better understand adaptation at a larger scale across the distribution of a species? So to build intuition on how we're thinking about this, we have to forget about a species distribution as a single uniform entity and think of it as a collection of smaller geographic distributions of genotypes or populations that might be more narrowly adapted to their local climates. So when the environment shifts, there might be different processes that we can observe and study or track, such as that there are already pre-adapted genotypes that do not suffer any extinction, that there might be some populations or genotypes that suffer extinction, and some populations might have been rescued by some gene flow and evolutionary rescue. And of course, all these patterns will be contingent on how natural selection or climate is changing locally, exerting that evolutionary pressure. And argue that genomic data sets are becoming comprehensive enough that we can start diving into these subspecies processes, as also Maria has tried with species within species trait variation. So today I'm going to address 3 questions related to this potential adaptive response of a species to climate change, two published studies and one unpublished one. So the first question is going to be, is there genetic variation to survive an extreme environmental event, such as those that we might expect with climate change? How exactly natural selection patterns are going to change locally? And finally, what phenotypes might be underlying adaptation or might be under selection? So let's go for the first. So to improve our understanding of potential evolutionary responses to climates, we're using the genetic model plant Arabidopsis thaliana as an eco-evo model. So Arabidopsis has the advantage that it's an annual plant, so we can study a whole life during a year, and we can measure lifetime fitness just by counting seeds at the end of the season. It lives all over the world almost, so we can capture big climatic gradients. The center of the distribution is around Central Europe, and the warm edge is in the Mediterranean. And I've observed it, I've photographed it flowering in the snow as well as in sand dunes. And recently we published the first 1,000 genomes of Arabidopsis from ecotypes collected all over the world. So to begin understanding the potential genomic basis of survival to an extreme event, I selected over 200 native populations of Arabidopsis from the 1,000 genomes and planted them in our greenhouse and started a terminal drought experiment to see how long they would survive this extreme event. To monitor whether they survive or how long, I use this image-based pipeline that would allow me to track when they died, and you can see that the green pixels disappear. To give you some intuition, there were plants that survived 4 weeks this drought, and some plants that survived 6 weeks. So I use this survival index as a phenotype that later put through a genome-wide association. So if you're not familiar with genome-wide association analysis, it's essentially a linear regression where you have in the y-axis, in the y, the survival, and in the x, particular variants at a position in the genome for which your plants might have either A or G, for example. And you compute this regression and do it iteratively for all the variants in the genome. In this case, 1.5 million polymorphisms, natural polymorphisms that these populations were different. And then a bunch of other correction factors. So overall, what we found was that about 50% of the survival variation could be explained by genetics. We found in this Manhattan plot that shows the significance of the association between each variant along the genome with survival. We found 150 variants that explain this variation, that were significantly explaining survival. And we found that survival was correlated with precipitation of origin, pointing out that these genetic variants probably cause local adaptation to precipitation. Okay, so this was not super surprising that there was some variation to surviving a climate extreme, but more interestingly to me was where these potentially adapted genotypes are. And to study this, I use a technique common in ecology, which is species distribution models or environmental distribution models. But instead of modeling the species distribution, I model the distribution of a strong allele, an allele that increases survival. So normally we have these geographic locations with a couple of labels, strong allele versus weak allele, and combine that with a climate map. We use a decision tree classifier, find the threshold in the climate where that strong allele is found, and we can project the distribution of the strong allele. And we can repeat this for every allele that we found or every genotype that we found to increase survival. Because survival, again, was a polygenic trait, so you need many alleles to survive it. And what this produced, by collapsing many of these distributions of drought survival alleles, we get this map of drought allele count. In darker areas are places where there are more of these drought alleles, and lighter areas where there are less. What we found is that those areas at the edge of the distribution, remember the Mediterranean is the warm edge, and the northern edge of the distribution is Scandinavia and eastern Russia. we find that there are more of these drought alleles, indicating that these populations might be already pre-adapted to extreme air conditions. You can imagine that if precipitation decreases in Europe towards the future, different populations might have very different susceptibilities to this decrease of precipitation driven by climate change. So we could potentially use this for conservation purposes in other species. Okay, so we've seen that there is indeed genetic variation to survive an extreme climate event, and that these genetic variants might be at the range edge. But we know that climate is not going to change all at once towards a big, massive droughts, but climate is going to change locally differently. So we need to understand how selection is changing across the landscape. And this is what I ask in the second part of the talk. So to study selection in nature, I conducted a common garden experiment in 2 locations with 500 Arabidopsis thaliana ecotypes from the 1000 genome. This is the location of Spain, which is again the warm edge of the distribution, and in another location in Germany, in the center of the distribution. A small historical parenthesis, this was actually inspired by one of the first common garden experiments ever conducted and published in AMNAT in 1941 by Klaus and Kekin Heisey. And interestingly, I am right now in my current institution, is the Carnegie institution at Stanford, and you can see the field site in the western side of campus. So I did a very similar experiment as Klaus and Kekin Heisey with some variation. I grew these plants, in this case is a picture of the foil tunnel in Germany, very similar to the one in Spain. So I planted 25,000 pots with Arabidopsis, and half of all the replicates, I water them under a Spanish-like rainfall, and the other half as a German-like rainfall, so high. And then over season, from fall to spring, I monitored who survived, and you can see how beautiful they become with anthocyanin accumulation when they're stressed. And I also measured fecundity by counting fruits automatically with an image pipeline. So what this produced is that by spring, I had lifetime fitness recorded for 500 genotypes in 2 locations in 2 rainfall treatments. And this is what I'll use to understand natural selection. Today, I'm going to talk about the treatments that made most sense or that are most natural: Spain and low rainfall, in Germany and high rainfall. So in you already can see that in Spain and low rainfall, actually there might be a strong selection because many genotypes have close to 0 fitness. But I wanted to understand this natural selection at the genetic level. So I use a genome-wide association approach, but instead of with a trait, directly with relative fitness. So the association that I would be calculating is a total selection coefficient over an allele. So if it's positive, that allele will increase in frequency. If it's negative, it will decrease in frequency in response to the selection. And again, we have in the y-axis the strength of the association and all the chromosomes in the x-axis. And at a glance, we see selection is highly polygenic. There are thousands of SNPs in the genome, thousands of variants in the genome that are under selection. They're expected to change as much as 25% in frequency in a single generation, so super strong selection. And in Germany and high rainfall, because the fitness differences were much smaller, the selection strength was weaker. We are expecting a slower turnover of allele frequencies, but also selected SNPs across all chromosomes, so also polygenic. This was not a surprise that in the warm edge of the distribution, selection was stronger. In fact, many genotypes completely died. But was selection in the same direction? So to study that, I bring this visualization where each dot in this scatter plot is one allele in the genome, over 1 million alleles. And they're mapped there in the x-axis by the annual precipitation in the origin of that allele. And we do that by cross-referencing the GPS locations of the 1000 genomes. And in the y-axis, precipitation seasonality. I also mark in black the average precipitation and seasonality of Spain, the location where we did the experiment. In color-coded are alleles, in green if they were positively selected in Spain, and in red if they were negatively selected. And what we see is that those alleles that were coming from lower precipitation environments were positively selected in Spain. And as they come from further and further in higher precipitation locations, they're more and more negatively selected. And we have the reversed or the mirror observation in Germany, that the alleles from high precipitations were positively selected in Germany. So what this is telling us is that there's a signature of local adaptation at the genetic level. Alleles are selected in their home environment, probably because our field experiments are replicating the same natural selection pressures that are occurring there. But this was very interesting because it's telling us that by knowing in what environment an allele has been observed, we can know in what environment it's going to be selected for. So I thought I'd formalize this with a random forest decision tree model, where we would try to predict total selection coefficient over an allele based on the climate where selection is taking place, the allele climate of origin, and some selection signals along the genome and functional information. So long story short, this model was thoroughly tested using cross-validation, hiding a whole chromosome, and trying to predict allele selection there. We also used other independent field experiments conducted by other colleagues across Europe and tried to validate that there was predictability. And in fact, there was between 30% and 80% predictability of allele selection based on climate of origin. But essentially, long story short, what we have is a very complex model that essentially is interpolating the selection coefficient of an allele across an environmental gradient. So could we predict what is going to be the selection in the future? We can do that because we have present maps and future climate maps from the Intergovernmental Panel for Climate Change. So what I did was to download the climate at all 500 locations where Arabidopsis lives nowadays. I feed those climate values to the random forest and predict what alleles are going to become more negatively selected or more positively selected by future climates. And then I checked at each of those populations, what is the allele that the plant there, the local plants have. So if they have many alleles that are going to be more negatively selected in the future, we can imagine that they are becoming more maladapted in the future. This is like the ancestry DNA or 23andMe predictions of cardiovascular disease from your genome, but in maladaptation. And similar as before, what this uncovered was some type of quadratic relationship with latitude. It was those populations in mid-latitudes that seem to become more maladapted in the future. And from the first question, we know that this might be because they don't have drought-related alleles. As you can imagine, this could be super useful if we had maps like this for threatened species to prioritize or to help assisted migration. Okay, so we found that natural selection is polygenic, is strong, it depends on or is recapitulated by local adaptation signals. But what might be the phenotypes under selection? This is finally what I want to ask, and this is unpublished work where we try to understand selection in the light of trait ecology. So my first step to measure selection over a key trait was that only one trait that we could really measure in Germany and Spain, which was flowering time. So I found an evidence of the classic escaping strategy of annual plants. If you flower earlier in the season, so left in the plots, more of the replicates remain. So viability selection was selected for earlier flowering. When I looked, however, at fecundity selection, there was a shy positive relationship between later flowering time and higher seed set in the low precipitation Spanish environment, potentially pointing at that there might be some trade-off between different traits that are under selection and correlate with flowering time. But it was impossible for us to really measure many more traits in those field experiments. So what we're doing to study multivariate phenotypic selection in Arabidopsis is using the large amount of already published Arabidopsis papers measuring a variety of traits. So in total, we're using 1,846 phenotypes, which we're classifying in 3 strategies related to coping with droughts. The first you already know, escaping. So those traits that were earlier flowering so that you don't experience drought before it comes, or dormancy of seeds. Avoidance, increasing water use efficiency by closing stomata or by gathering more water with a better root architecture. Or tolerance, tolerating desiccation, which might not really properly exist in Arabidopsis as in mosses or other things, but we can categorize metabolites that can act as osmolytes to stabilize cell and tissues. So with all these traits and using machine learning methods for imputation, we now get a database of over 1,000, almost 1,000 traits for 2,000 accessions of Arabidopsis, which are all these dots. And we decompose this variation using principal component analysis, which captures around 45% of the variation, the 2 first axes, which I think is a lot. So now if I lay on top of this the loadings of phenotypes in this species, what we find is that the phenotypic scape of Arabidopsis is scape dominated. So in the left, we have late flowering plants, and the right, we have early flowering plants. And the second axis is separating 2 types of early flowering plants, the woody Arabidopsis that we use in laboratory in genetics, and the Mediterranean fast cyclers and bed hedgers. And I recommend you check out a paper from Martina Verdeja where they discuss these combinations of strategies along with a commentary from mine. But so the first species is interesting because it not only accumulates variation in flowering time, but also an array of phenotypes was correlated with it. So with late flowering time, we have slow growth, large plants, high water use efficient, and this is often measured with delta carbon 13, that means stomata have closed. Higher stomatal density, survival in my drought experiment in the first question, which was also included in this analysis. And on the other hand, we have early flowering plants, rapid growers, low water use efficiency. And this makes sense, but from our experiment in Spain and low precipitation, early flowering was selected. So low water use efficiency might have been selected. How can this be? That seems counterintuitive. So to try to shed some light into this phenotypic selection and correlated traits, I use the classic Lande and Arnold approach, where you can compute selection over phenotypes, accounting for covariation between traits. And indeed, we found that there was selection for earlier flowering and lower water use efficiency, delta 13, but only if we don't account for the covariation. So once the covariation between water use efficiency and flowering time was accounted for, the gradient of selection over water use efficiency was not significant. So this might be telling us that low water use efficiency in Spain, low precipitation was only selected because of a correlation with early flowering, and might be pointing at some type of constraint between escape and avoidance. But this might be due to not having enough power or because we didn't measure enough phenotypes. So I wanted to go more comprehensive and repeated this multivariate selection analysis with 22 species that accumulate 95% of the variation in Arabidopsis thaliana. And what I found was, again, evidence for selection of early flowering and low water use efficiency in Spain, but also, interestingly, PC19, a lost species that only explains 1%, was positively selected. And I saw the loadings were dominated by water use efficiency. So this is interesting. Selection is actually trying to maximize both avoidance and escape, but because of the anti-correlation between avoidance and escape, it might not be able to do so. So why there is an anti-correlation between escape and avoidance? There might be 2 options: is environmental or is genetic. Environmental would mean that perhaps the water use efficiency trait delta carbon 13 was not measured in the field experiment, and the rank orders changed, and all my selection analysis don't make any sense. If it's genetic, it will mean that there might be a constraint. because we have whole genome data, we can actually measure genetic correlations between two traits. So I measured heritability of delta carbon 13 and flowering time in their genetic correlation. And indeed, they were positively correlated. So there's a trade-off escape avoidance that is genetically based. And again, using Landy and Arnold, you can see that lower water use efficiency would evolve after selection in Spain, which again is counterintuitive. And repeating delta, the analysis of changing phenotype, the response to selection on PC19, which remember was the delta 13 that is flowering independent, it's going in the opposite direction. So again, I think we're seeing here a conflict of selection trying to maximize escape and avoidance, but they're anti-correlated, and this has a genetic basis. Now, so if it has a genetic basis, it could be due to two things that are pretty relevant in disentangling this selection. The first could be that there are two loci that independently are coding for flowering time and delta carbon 13 water use efficiency, and selection has put them together in linkage disequilibrium, so the two traits correlate. But with enough recombination, if selection was trying to maximize escape and avoidance, it would manage because they can become unlinked and have independent responses. But if there is a physiological constraint or a genetic pleiotropy constraint, a true constraint, you couldn't maximize both. And this might be the case, for example, of a locus that is a transcription factor that is expressing or controlling expression of multiple pathways that have pleiotropic effects into traits. So finally, to try to disentangle linkage and pleiotropy, I use a new GWA approach called Bayesian sparse linear mixed model that should correct for linkage effects. And I use PC1 that was escape and PC19 that was avoidance, and are uncorrelated by definition. They are orthogonal PCs. So that if in the GWA I find the same allele affecting both traits, then it has to be pleiotropy. So what we have here is the top alleles in both GWAs, and we have the effect of an allele in escaping in the x-axis and in avoiding in the y-axis. What we find is that those alleles increasing water use efficiency in the y-axis, these top ones, are negatively selected in Spain. This could be reminiscent of some covariation, but the annotation of these genes are actually indicating us that is a bona fide water use efficiency genes that are triggering a stress signal, which you can imagine is pleiotropic by starting a syndrome of arresting growth but increasing resilience. So definitely seems that there's a genetic pleiotropic constraint to selection over water use efficiency that persists at the allele level. So will climate change lead in these populations to early flowering or water use efficiency? To be continued, so stay tuned. So to recap, we found signals of phenotypic selection for the classic escape strategy, and it seems that perhaps also avoidance, although there's a constraint. Note that these genetic constraints on different relevant ecological traits are perfect raw material for machine learning models, like in the second project, to predict selection in different climates. So what's next? Two areas that I'm very excited for the future. One is studying allelic selection and its interaction with demographic dynamics leading to extinction or evolutionary rescue in a global evolution experiment with Arabidopsis. And the second is to really tease apart those pleiotropic and complex mutational effects by using gene editing in Arabidopsis in the lab. So finally, I want to thank all my supporters and funders, colleagues collaborating in my group, and all of you for your attention. Thank you.
Thank you very much. We have a couple minutes for questions here. Let's see if any come in. I have a few if nobody else has any. Yeah, so I guess one of my questions is, I'm always struck by these studies looking at I guess the question I was wondering is, based on the climate modeling, is there ever going to come a time when the change in the climate completely exceeds all of the variation, like the maximum drought tolerant genotype that's there, or the maximum wet tolerant? Is climate change going to push it so that there's simply nothing there, and you can predict when the population will go extinct?
Absolutely. That's, I think, a great question. For sure, there will be a extreme that there is no enough genetic variation and not enough generations for those populations to adapt to that. And we don't know where that extreme is. The previous, this previous slide, the global evolution experiment, might give us some clues about this, because we're conducting this experiment in 45 sites, and we're already seeing that a number of sites these experimental populations are disappearing. So that will be perfect for us to understand when local extinction is happening, what extreme environments are leading to that local extinction, and whether there's really a limit to adaptation. For sure.
There are two more questions, but I think we're out of time. There's one on Slack, and then there's two more in the chat. So yeah.
Okay, I'll answer them. Thank you.
Thank you for that great talk, and let's have a round of applause for Moises.